Okay, welcome back students. Today we are going to discuss unit 5 and in this particular unit we are going to discuss sexual reproduction in flowering plant part 2. Before I start the main content, I would like to inform that the images used in this slide are for education purpose only. We respect intellectual property right of the original creator and owner of these images. If you are the owner of any of the images used in this PPT, have any concern, any issue, please contact us at sathee@iitk.ac.in and we shall remove the images. In part 1, we have already discussed about the structure and function of flowering, different part of flower, male gametophyte, female gametophyte, and in this particular part 2 section, we are going to discuss about pollination, type of pollination, agents used in pollination, okay, fertilization, development of endosperm, embryo, seed, fruit, differentiation, and organ formation. Okay, so these are the highlighted points which we are going to discuss in this particular video lecture. So, what is pollination? As you know that both male and female gametes are non-motile in plants, right? And they need to brought together for fertilization. Why they are present in the flower? Because of the fertilization, right? But they are non-motile, right? But with the help of any agent, any abiotic agent or biotic agent, they need to be brought together for fertilization, right? So, transfer of pollen grain from the anther to the stigma of the pistil is necessary. So, to achieving this target, there are various kind of adaptations are there in flower, okay, and flower also use certain kind of external agents for this pollination point of view. Okay, so let's divide or let's discuss different type of pollination. First type is autogamy, then getonogamy, and then third type is xenogamy. Autogamy, getonogamy, and xenogamy. Okay, when self-pollination occur, means transfer of male anther in the same flower, okay, to the female stigma part, that time we can use the term self-pollination or autogamy. Clear? When transfer of anther of one flower to the stigma of another flower of the same plant, that time we can use the term getonogamy. Transfer of anther from one flower to the neighboring flower of the same plant occur. That time getonogamy. And when transfer of anther occur from one flower of a plant to the stigma of another flower of some other plant, same plant but another plant, not the self plant, that time we can use the term xenogamy. Okay, so one by one let's understand all three terms. So autogamy, pollination, when pollination occur within the same flower. Okay, so that time we can use the term autogamy for the same, and this autogamy is rare in normal flower due to the need for synchrony in pollen release and stigma perceptibility. Because of this one is rare. There are two type of flowers. One is known as chesmogamous flower. Another is known as clastogamous flower. So chesmogamous flower are like this, means open flower with exposed stigma, exposed anther and stigma. This type is known as chesmogamous flower. Another type of flower are like this, which are not comes under open flower category. so they are not open. And in this flower, they ensure self pollination and assure seed set without pollinator. They don't need any kind of pollinator agent. Okay, but the susceptibility or perceptibility, if time is differ, okay, pollen release time is something different than stigma perceptibility. That time normal flower also not follow this autogamy type of pollination. So it is rare in normal flower. Second one is geitonogamy type of pollination, means transfer of pollen from the anther to the stigma of another flower on the same plant. Plant is same but flower are different, right? So transfer of anther to the stigma of another flower on the same plant is known as geitonogamy. So functionally this is known as cross pollination, but genetically similar because of the presence of same plant. Genetically similar to orthogamy because plant is same, just flower is different. Okay, so functionally we can say cross pollination occur, but genetically it is similar to orthogamy. Clear? Third type is xenogamy. So transfer of pollen from the anther to the stigma of different flower. So these are genetically different. Okay, so the only type, this xenogamy is the only type that brings genetically different pollen to the stigma. Xenogamy. Okay, I hope all three terms has been cleared now. So now let's understand different agents which are useful in pollination. Some abiotic agents and some comes under biotic category. Abiotic means wind, water, these type of agents comes under abiotic agents category, while if some kind of animal, insects are involved, that time biotic agents, right? So first is abiotic, and in abiotic first type of pollinator is agent is wind, and this one is very common among abiotic pollination. What are the characteristic? For to transfer the pollen grain through wind, pollen grain should be light in weight. These must be non-sticky. Okay, and in that case stamen should be well exposed. Okay, sometime pollen grain are large, or and stamen also large, and pollen grains sometimes have feathers, so they can very easily move from one end to another end. So feathery stigma. Okay, large and feathery stigma, well exposed stamen, so that easily they can interact with each other. So pollen grain should be light, non-sticky, stamen are a well exposed form, and stigma large, so very easily pollen grain can settle down there, and it should be feathery. What are those examples? Examples are corn and grasses. The flower, as you can see, this flower of wind pollinator flowering plants such as this sawtooth oak, Quercus acutissima, are less showy than insect pollinator flower because they don't need to attract insect because of their pleasant smell and colorful petals. They just utilize wind as a pollinator agent, so they don't need any kind of attractive petals or scent type of smell. Okay, so but what are the main thing? Well exposed stamen and large feathery stigma and light and non-sticky pollen grains. Some pollens which are present in kind of plant which are present in the water use water as a pollinating agent. Okay, and this water pollination is very uncommon or rare in flowering plant. Okay, And these are common in some lower plants like algae, bryophytes and pteridophytes. They utilize water as a pollination agent. Okay, what are those examples? Vallisneria, Hydrilla, Zostera. These are the examples of lower plants which utilize water as a pollination agent. In this case, female flower reach the water surface. This is the flower, reach the water surface, and male flower or pollen float towards the stigma. This is the main flower. So through floating, the pollen float towards the female flower, and then fertilization occur. Pollen grain offer have these pollen grain have a mucilaginous covering to prevent wetting. Okay, because they are present in the plenty of water are there, they quickly wet it down. Okay, so to protect its outer surface, its viability, pollen grains in these type of plants, lower which are present, lower plants have mucilaginous covering in the outer surface. Now, biotic agent means animal pollination. All those plants which utilize animal or insects for their pollination purpose. And this animal pollination is very common in flowering plant. What are those common agents? Bees, butterfly, flies, beetle, wasp, ant, moth, bird, hummingbird, bat. Some primates also like rodents, reptiles can also transfer pollen grains from one plant to another plant. Okay. Now, what are the characteristics of animal pollination? Flower are large, colorful, attractive. It should have fragrant, a kind of specific fragment, and nectar rich. These are certain characteristics which actually attract animals or insect towards towards their end, towards their flower side. Okay. But sometimes flower emit a foul smell, also foul odor, to attract specific pollinator like flies and beetles. Example, Amorphophallus. This is the example. Amorphophallus. They provide a safe place for insect to lay egg. And Yakka and moth. Both of them are mutually dependent to each other to complete their life cycle. Okay. Now let's discuss some outbreeding devices. So these outbreeding devices actually prevent self-pollination and encourage cross-pollination to avoid inbreeding depression. If continuously self-pollination occur or self-fertilization occur, then after some generation, after 4 or 5 generation, inbreeding depression comes, in which recessive character get showing it up, and it will reduce the what is the right term for that? It will reduce the acceptability and prominent property of that plant, or in also in animal case, right? So to avoid the inbreeding depression, sorry, and to cell pollination must be prevented. And for this, to encourage cross pollination, some kind of outbreeding device has been used up by the people. Okay, so temporal separation is one method. Means pollen release and stigma perceptibility are not synchronized. Means time of pollen release and time of stigma perceptibility are not same. So if both the things showing their maturing behavior or releasing time of pollen and perceptibility time of stigma is different, then obviously it will promote it will discourage self pollination, right? Second type is spatial separation. Means anther and stigma positioned to prevent contact within same flower. The position of anther and stigma is like this, that they cannot transfer of pollen grain to the same stigma is not possible. So their positioning are different. So anther and stigma positioned in such a way to prevent the contact within the same flower. It is known as spatial separation. And the release time of pollen and stigma perceptibility time is different. That time we can use the term temporal separation. Third type is self-incompatibility. This one is the first, followed by second and third. Self-incompatibility. Genetic mechanism. It is a kind of genetic mechanism which inhibit self-pollen germination or pollen tube growth. Okay, it is a genetic mechanism which inhibits self-pollen germination and pollen tube growth. There are two types of flower. In unisexual flower, there are two types of flower, monoecious and dioecious. Monoecious means both male and female flower are present on the same plant. Like castor and maize, it prevent autogamy. Okay, in dioecious, male and female flower present on different plant. Like in papaya, it prevent both autogamy and gytonogamy. So in unisexual flower. Now let's discuss pollen-pistil interaction. Means fertilization. This is the first step. For pollen-pistil interaction means transfer of pollen does not ensure compatible pollen. So pistil must recognize it and accept it or reject it. It depends upon the pistil. Okay. So just transferring of pollen to the stigma site is not we cannot say that fertilization 100% occur. Actually, pistil must recognize that kind of pollen is healthy or not, or whatever the condition it is required. So pistil must recognize the right pollen, and it depends upon the pistil to accept it or reject it. Okay, if pollen are compatible, it is accepted and it will promote fertilization. If pollen is incompatible, that time it will reject by the pistil. How? Just to blocking germination or growth. Clear? Now the process of pollen pistil interaction, it involves certain kind of chemicals. Certain kind of chemical give a specific signal for to proceed fertilization. Okay, so compatible pollen germinate on the stigma. There are so many pollens come and just landed on the this stigma part, but only the compatible 1, 2, 3, 4, 5, 6. Only the compatible pollen germinate on a stigma, forming a pollen tube. So here fourth number is stigma found this one compatible. So this pollen is allowed by the stigma to germinate or grow pollen tube towards the ovary side. Right? So pollen tube enter to the ovule like this. Okay, through this region, and this region is known as micropylar region and synergids. This is the synergids and this is the micropylar region. Okay, now artificial hybridization. Artificial hybridization is used up by the breeders. Okay, so breeders actually cross species, different species to develop better variety. And for that, they use various kind of technique. First one is emasculation. Emasculation means removal of anther from bisexual flower. Just removing these anther from the bisexual flower. So the same kind of anther will not during pollination transfer to the stigma of the same flower. Okay, so this is known as emasculation. Another technique is bagging. Means this is the bagging. Means covering the flower to prevent contamination by unwanted pollen. Just to promote self-pollination. Okay, unwanted pollen doesn't come through wind and fertilization occur. So cover the flower to prevent unwanted pollen to come and get start fertilizing that particular oval. Right. So pollination with the desired or specific quality pollen when stigma is perceptive. Okay, just cover the flower and pollinate that particular flower with the specific pollen when that particular stigma is perceptive. Now let's discuss double fertilization. As after pollen pistil interaction, out of so many pollen grains, only the compatible pollen is going to allow to develop pollen tube. Okay, as the compatible one is starting developing pollen tube, like in this picture, in this one, it enter a synergids from this end and releasing two male gametes. Releasing two, as you know, as you can understand this is a pollen tube. And two male gametes are there. One is generative cell, another tube cell. So first step, initially the pollen tube consists of two haploid cell, right? The generative and the tube cell, right? So syngamy and triple fusion, there are two terms which is related to this double fertilization. Syngamy means one male gamete. In pollen tube, two male gametes are there. So one male gamete fused with the egg cell. One male gamete is fused with the egg cell. One sperm cell fertilized with the egg cell and form diploid zygote, 2N zygote. While the another one, other male gamete fused with the two polar nuclei which is present in the central cell. This is the central cell. So fusion of this second male gamete to the two polar nuclei which is present in the central cell. And this process forming a triploidy primary endosperm nucleus. PEN, primary endosperm nucleus. This. So double fertilization occur. First fertilization when one male gamete fused with the egg cell over here, and another male gamete fused with the polar nuclei which is present in the central cell. So that's why it is known as double fertilization, as it consists of syngamy and triple fusion. And this property, double fertilization, is unique property of flowering plant. Clear? Now development, the center part, this one is the center part, right? Central cell become convert into the primary endosperm cell, PEC. This is known as PEC, primary endosperm cell, central cell, and develop into the endosperm. Okay, and develop into the endosperm and zygote. This is the zygote. Zygote develop into the embryo. This is the zygote develop into the embryo. Okay. Now let's discuss post-fertilization events. It include endosperm development and embryo development both. It include post-fertilization event, endosperm, embryo development and ovule maturation into the seed and ovary maturation into the fruit. Okay, so ovary mature into the fruit while ovule mature as seed after fertilization. So first endosperm development, it precede embryo development, then primary endosperm cell divide to form triploid endosperm tissue. It provide nutrition to the developing embryo. Which one provide nutrition to the developing embryo? Endosperm. Clear? Now, the free nucleus endosperms are also present there. This PEN, primary endosperm nucleus, undergo a nuclear division to form free nuclei, so many free nuclei. If cell wall is formed, it will make endosperm cellular. Like in coconut water comes under free nuclear endosperm category, while that hard part, white kernel part, comes under cellular endosperm category. And it's a very important line. Sometime question asked on this particular line. Okay, so fully endosperm, that liquid and hard part fully consumed, by us, nariyal water, nariyal white part of the nariyal, right? So this fully consumed by the developing embryo before and seed maturation. Okay, like pea, groundnut, bees. And sometime this endosperm persists in mature seed and used during germination, like in castor, coconut. Now, embryo development. This embryo development occur at the micropylar site of the embryo sac where zygote is located. Zygote divide after some endosperm formation and to ensure nutrition. Earlier embryogeny stage are similar in both monocot and dicot. So first dicot embryo, let's discuss dicot one first. So it consists of embryonal axis and two cotyledon. These two are cotyledons. Okay, and this is the axis part. So epicot, this arc epicot means part above the cotyledon comes under epicot category. It ends with the plumule here. It ends with the plumule means stem tip. Right? Hypocotyledon part just below the cotyledon. This. Just below the cotyledon, this comes under hypocotyledon category, which ends with the radicle, which is covered by the root cap. Clear? Now, monocotyledon embryo. This monocotyledon embryo have only one cotyledon, which is called scutum. Only one cotyledon, which is called scutum. It is situated laterally. Radicle and root cap. This is the radicle and root cap. This region is enclosed in a sheath known as coleorhiza. This is the sheath. This is known as coleorhiza. Right? Okay. Now, epicotyle. Epicotyle is present just above the scutum. It is having shoot apex and leaf primordia enclosed in coleoptile here. Okay. Now seed formation. Seed are the final product of sexual reproduction. Seeds are the final product of sexual reproduction in angiosperm or flowering plant, and these are formed inside the root. Right? So it consists of, this seed consists of the outer surface, seed coat, and then two cotyledons are there and one embryo sac. So it is consist of seed coat, cotyledon and embryo sac. Cotyledons are thick and swollen due to storage of food reserve. Mature seed can be non-albuminous, means there is no residual endosperm, or albuminous if endosperm are there. Right? So mature seed we can classify it into two categories, non-albuminous or albuminous. Seed coat is hard. It is having hard covering, which is known as hardened integuments of the ovule. This hard part. Right? Micropyle remain present as a small hole. What is the need? Just to supply of water and oxygen. So it is act as a entry point for oxygen and water, which is required during germination time. Okay. Now seed maturity. So this is the outer covering seed coat, then this is endosperm, and these are the cotyledons. Okay, this is the hypocotyledon part, root axis, and this is the shoot apical part. Now seed maturity. When seed get mature, water content get reduced and become dry. Okay, so embryo enter in the dormancy form, or it germinate only under favorable conditions. As you can see over here, this is the another picture of a seed. This the pink line is seed coat, and this is the cotyledon. Okay, this is the shoot apex, this is the root apex, and this is the endosperm site. So after maturing, seed enter into the dormancy side, and it only able to germinate under unfavorable conditions. Now fruit formation means ovary develop into the fruit. ये तो हमें पता ही है, ovary develops into the fruit simultaneously with seed formation. Pericarp form ovary wall. Fruit can be fleshy or it can be dry, and it may have dispersal mechanism. True fruits develop from the ovary only. If true fruits develop from the ovary, but some fruits comes under false fruit category. All those fruits which involve other floral part like apple. This apple comes under false fruit category, and so many questions asked in various competitive examination with this example, using this example. So false fruit involve other floral part. Okay, a term known as parthenocarpy or parthenocarpic fruit. These fruit develop without fertilization. There is no fertilization occur in parthenocarpy. So the type of food is without seed. So seedless fruit has been developed because there is no fertilization. Example, banana. Okay. Now what are the advantages of seed? Seeds are independent of water for reproduction. Aids in dispersal and colonization, provide nourishment to seedling. Seeds provide protection by hard seed coat, and it generate genetic variation. Seed viability. Some seed lose viability within few months. Some seeds can remain alive for hundreds of years. So it depends upon the variety of the plant. Okay. Some oldest viable seeds are discussed over here. One of the example is lupine. Lupinus arcticus. It germinate after 10,000 years, and these are found in Arctic tundra region. Another example is date palm. Phonis dactylifera. It germinate after 2000 years, and these seeds are found at King's Herod's palace near Dead Sea. So these are certain example of seeds which are still having viability in it. Okay. Now a term known as apomixis is also need to discuss over here. Means if the seeds produce without fertilization, some kind of seeds are having ability to produce without fertilization in sub-flowering part, that time with the help of this apomixing technique. What are those example? Asteraceae and grasses. In these type of plants, in this type of category, seeds are able to produce without fertilization. As this apomixis is a type of asexual reproduction, but this process just mimic the sexual reproduction process. Okay, this apomixis can occur via diploid egg cell forming without reductional division and developing into the embryo without fertilization. What is apomixis? Apomixis is a technique which can occur via diploid egg cell forming without reduction division and developing into the embryo without fertilization. It often involves nucellular cell dividing and developing into the embryo, and this one is common in citrus and mango. These are the examples of apomixis. Another term which is also related to today's topic is polyembryony. So polyembryony means occurrence of more than one embryo. Occurrence of more than one embryo in a seed is known as polyembryony. 1, 2, 3, 4, 5, 6. More than one embryo within a seed is known as polyembryony. Okay, this is a single embryo is there, monoembryony. If more than one seeds are there, more than one embryo are there, then we can use the term polyembryony. Okay, so it we can see this polyembryony in some species like oranges, where seeds contain multiple embryos of different size and shape. Now, what is the genetics of apomictic embryo? Apomictic embryo can be considered clones, as they do not undergo genetic recombination, but they are similar kind of embryos, but there is no genetic recombination. What is the importance of this type of embryo in agriculture? If we develop a kind of hybrid variety, it will increase productivity but require annual seed production. Apomixis could allow hybrid to maintain their characteristic without segregation, and it reducing the cost for farmer. Some active research is also going on this particular topic, which transfer apomictic gene into the hybrid variety for sustainable crop production. Okay, so these are some points related to this today's video lecture. I hope most of the things has been cleared now. Now, with part 2, I have completed sexual reproduction in flowering plant topic. I hope most of the things has been cleared now. Still, you have any doubt, any clarification, any question, please let me know. We will discuss it later on. Till then, take care, bye bye, and keep learning. Namaskar.